or personal data or anything like that.
Okay, thank you. So I don't have any other questions at the moment from Alana. So if Ed is here and ready to get going slightly earlier, then I think we'll do that. Thank you very much, Alana, and you can stop sharing your slides now.
Sure, thanks so much for having me. Thanks a lot.
So I'm looking around to see whether I can see.
So Ed has just joined. Might take a few minutes for him to get set up, but he has joined the meeting.
Thank you.
Hi, Rowena, can you hear me?
Yes, great.
Hi.
I can see you and hear you now. So
Fantastic.
sorry to get you off the starting blocks very quickly, but welcome. And yes, just to introduce you, I'm sure a lot of people have seen you before, but Dr. Edwin Jabbari, postdoctoral clinical researcher at the UCL Queen's Square Institute, has been involved with the PSPA over a number of years. And yes, exciting to hear what you're going to talk to us about today, which is development of a tau seeding assay as an early diagnostic test.
Great. Thank you so much for the introduction. I'll just share my screen. Can you confirm that you can just...
Yes.
Slides? Fantastic. Okay. So, yeah, so thank you very much for the introduction. I'm really happy to provide an update on my latest work that's been funded by the PSP Association. It's great to see so many participants in the audience. So I'm going to be focusing on one specific area today, which is the lack of tests that are available to accurately diagnose PSP and CBD patients in the early stages of the condition. And this is clearly an unmet need for many reasons. The first one is that diagnostic uncertainty and misdiagnosis is a really frustrating experience for patients and their families to go through. And I base this on my experience of looking after PSP and CBD patients for the last 10 years of my career. And it's such a well-trodden path for patients and their families to say that they were initially diagnosed with Parkinson's disease, and then later down the line, when things were at a much more advanced stage with the diagnosis, then at that point changed to PSP or CBD. So that's the first reason why this is important. The second reason is that misdiagnosis impacts greatly on the accuracy of results in research studies as well as clinical trials. So we as clinicians are very invested in getting the diagnosis right. And the third one, which is linked to that second point, is that the recent success that we've seen in Alzheimer's disease trials has provided, really hard evidence that when you have an effective treatment, the earlier you give that treatment, the better the outcome. So these are a number of reasons as to why it's important to tackle the problem of getting the diagnosis right at an early stage in PSP and CBD. So in the field of neurology, we place a really big emphasis on the importance of taking a history and examining a patient to make a diagnosis. And it is definitely important, there's no doubting that. However, for decades, we've known that relying solely on this, the history and the examination, to diagnose PSP accurately is very challenging. So the only way to say with absolute certainty that someone has PSP or CBD is to study their brain at post-mortem. And this diagnostic challenge has been highlighted in the past by clinicopathological studies, where it's been shown that the accuracy of a diagnosis of PSP made during life is around 80%. Now, looking at it the other way, when you take a cohort of patients diagnosed with Parkinson's disease during life and you follow them up over time, as shown by the campaign study here that I'm highlighting with my cursor, a handful of those patients who have been diagnosed with Parkinson's disease will be re-diagnosed as having PSP. Now, I think that part of the explanation is that for many years, we didn't know that PSP can present in a variety of ways, not just this classical form of PSP with problems with eye movement, problems with balance and falling over. There are alternative ways that PSP can present. And some of these alternative forms can look a lot like Parkinson's disease in the early stages. And raising awareness of these alternative forms of PSP is what we've been doing for the last few years in the Prospect study. So the knock-on effect of this is that overall, nowadays, we're better at diagnosing PSP, but the delays in diagnosis still remain a major problem. And our own data from the Prospect study has shown that the diagnostic latency, so the gap between patients presenting with symptoms and then finally receiving a diagnosis of PSP in that classical form of PSP that we know so much about, there are delays of up to 3 years. And then when you look at those alternative forms of PSP, the diagnostic latency can be up to even 4.5 years. So clearly we could be doing much better than this, and we should be doing much better than this. Now, to understand the past and present approaches to tackling this issue of diagnosis, I need to briefly outline what's going on in the brain of patients with PSP and CBD. And it's all to do with tau protein. So tau is a protein that we all have in our brains, but for various reasons in the setting of PSP and CBD, the amount of this 4-repeat tau, which is a specific form of tau, the amount of that 4-repeat tau in the brain increases, and it becomes more sticky, and it clumps together to form these aggregates that you can see here highlighted by my cursor. And these aggregates are toxic to brain cells, eventually causing them to die. And these toxic forms of tau are able to spread from cell to cell in a predictable manner. And that causes healthy tau in the neighboring cell to become more sticky and abnormal. And this vicious cycle essentially is driving the progression of the condition, and it's causing the pathology to spread through the brain. Oh, and I should add that tau pathology is not exclusive to PSP and CBD. It's also seen as part of the pathology of Alzheimer's disease. Now, in Alzheimer's, there's a really accurate diagnostic test which begins to be abnormal even before patients have symptoms. And the levels of that test continue to rise as the disease progresses and becomes more symptomatic. And this test is the measurement of specific fragments of the tau protein in blood or spinal fluid. So you'd think that the same logic would apply to samples from PSP and CBD patients. But what you'll see here, if you look at this non-Alzheimer's disease group highlighted by my cursor, the levels of these, if we measure the levels of this tau protein, these fragments of tau protein, they're exactly the same as what you would find in someone that's neurologically normal, has no neurological symptoms. Exactly the same if we compare these two groups here on the far right and the far left. And this non-Alzheimer's group contains samples from PSP and CBD patients. So this hasn't proven to be the case in PSP and CBD, and there are ongoing efforts to try to understand why that's the case. Similarly, there was a lot of interest in the measurement of neurofilament light chain levels in blood and CSF, because the levels in PSP and CBD are raised, as you can see from this diagram here. But the problem is that it's also raised in a number of other neurodegenerative conditions. So it's not helpful as a specific diagnostic test for PSP and CBD. So the take-home message is that up until now, we've not had any promising diagnostic blood or spinal fluid tests for PSP and CBD. So this is where I think we're heading in terms of a solution: seed amplification assays. So I want you to cast your mind back to a few minutes ago when I said that in PSP and CBD, tau becomes sticky and abnormal, and that abnormal tau can subsequently spread and cause tau in neighboring cells to become abnormal. So with these tests, these seed amplification tests, essentially what you're doing is you're replicating that process in a test tube. And this is how we do it. So you take man-made synthetic tau that's created in a laboratory, and you put that synthetic tau in a test tube. You then add your patient's body fluid sample, so for example, spinal fluid samples, and that spinal fluid should contain abnormal toxic forms of tau protein, which in this case we refer to as seeds. Now, when the abnormal toxic forms of tau protein mix with the normal man-made synthetic tau over the course of 24 hours, it causes that synthetic tau to become more sticky and abnormal and clump together to form these aggregates. And the formation of these aggregates can be detected by adding a fluorescent dye to the test tube that allows us to visualize that process of aggregation. So I want you to look at this plot here on the far left. So you can see that there's an initial lag phase, and this lag phase is where we've added the patient's sample, which contains that abnormal toxic tau seeds, and they're mixing with the synthetic tau. And then all of a sudden, there's a big amplification in the signal of fluorescence. And that amplification is where there's aggregation that's occurring in the test tube, causing that synthetic tau to clump together and become abnormal. And that confirms the presence of abnormal tau seeds in your patient's biofluid sample. Now, there's been a lot of excitement in the Parkinson's disease world because many different research groups and now companies have used this technique to develop effective seed amplification tests to detect the presence of alpha-synuclein pathology. That's the pathology that's specific to Parkinson's disease, in contrast to tau in PSP and CBD. And these tests are now so advanced that they could potentially be rolled out as an NHS diagnostic test for Parkinson's disease imminently. So this is really quite advanced in its development. So the obvious question is, can we develop a tau seed amplification assay for the diagnosis of PSP and CBD? So I was really chuffed to receive a grant from the PSP Association that allowed me to travel to the US in February to spend a month at a laboratory whose prime focus is on the development of these assays, these seed amplification assays. And now in the last part of this talk, I'm going to share the preliminary results from that trip. So the aim here was to test both PSP and Parkinson's disease spinal fluid samples on their established alpha-synuclein assay, that's that Parkinson's assay, and then work also on developing a functioning tau assay, and then again applying both the PSP and Parkinson's disease samples to that tau assay. And remember that these patients have been diagnosed with PSP and Parkinson's disease based purely on a clinical examination, as we would normally do in a clinical setting. And of course, the ultimate aim is to bring this technique back to the UK to get it set up as a diagnostic test that we can offer to patients across the UK. So that was the aim. And what one might expect to see is that with the alpha-synuclein assay, all of the Parkinson's disease samples will be positive and all of the PSP samples will be negative. And then the complete opposite for the 4-repeat tau assay. We would expect all of the PSP samples to be positive and all of the Parkinson's samples to be negative. So we avoid this problem of misdiagnosis. So first, what we did was we applied the established alpha-synuclein Parkinson's assay to both our Parkinson's and PSP samples. And I'll just orientate you to the various plots that I've put on this slide. So on the left, on the top and bottom on the far left-hand side are our controls. So IR27 is a negative control. So this is spinal fluid sample from someone that we definitely know doesn't have any pathology in their brain. And on the bottom, labeled as PDCSF, is a positive control. So this is spinal fluid from someone that we definitely know has got alpha-synuclein pathology, Parkinson's pathology in their brain. So in the middle here is an example of what we saw in the majority of our PSP samples, a completely flat line, indicating that there's no alpha-synuclein pathology in the brain, which is what we would expect. In contrast, in the top right, you'll see an example of what we saw in the majority of our Parkinson's disease samples, a clear positive. There's no doubt that this is a positive. This patient has alpha-synuclein pathology in their nervous system. And then, interestingly, there was this small subset, these two plots here on the far right, in the middle, and at the bottom, this small subset where we got weakly positive results for the alpha-synuclein test. And interestingly, we saw this in both a small number of PSP and Parkinson's disease spinal fluid samples. So in terms of the actual numbers, we found that around 8% of PSP samples were weakly alpha-synuclein positive. And interestingly, these patients tended to be older and they progressed a little faster compared to patients who were alpha-synuclein negative. But remember that these are very small numbers of samples, so we need to see if this pattern is observed in a larger cohort. So the big question is, have we got the diagnosis wrong in these PSP patients who are weakly alpha-synuclein positive? Well, we don't think that's the case. Instead, what we think is that if we had a tau assay, these samples would be strongly positive, which was what we would expect in PSP. And in fact, this weakly positive alpha-synuclein result represents co-pathology, so a secondary low-level alpha-synuclein pathology, which might be impacting on the disease course in PSP. It might be making the disease progress slightly quicker. Now, in contrast, you can see that, as expected, 95% of the Parkinson's disease samples were alpha-synuclein positive. But interestingly, this small subset who were weakly positive, much like those weakly positive PSP samples, this begs the question, could these weakly positive samples be from patients who have been misdiagnosed with Parkinson's disease, and instead they might actually have primary PSP pathology? And this further highlights the need to develop a tau assay so that patients who we see in the clinic, their samples are run on both an alpha-synuclein assay as well as a tau assay to give us an accurate diagnosis of what's going on. So where have we got to with the development of a tau assay? So we worked very hard on this, and eventually we have been able to develop a 4-repeat tau assay, which is a very exciting development. Now, the starting point here is not the testing of spinal fluid. It's actually on brain tissue from patients who we can say with absolute certainty had PSP pathology in their brain. And you can see that in all 6, going down 1, 2, 3, 4, 5, 6, all 6 of these separate PSP brain tissue samples, we're getting strongly positive results using the 4-repeat tau assay, which is very promising. And similarly, when we applied the assay, the 4-repeat tau assay, to brain tissue samples from brain donors who had no symptoms of neurological disease during their life, we see no signal at all, as we would expect, apart from, interestingly, this last sample at the bottom, which is weakly positive for the 4-repeat tau. So when we saw this result, we were a bit worried that this was a spurious result that suggested that the assay might not be working as well as we would have hoped. It might not be as specific as we would have hoped. So we asked our neuropathologist to go back and examine the brain of this donor, this healthy donor. And interestingly, under the microscope, they found the presence of very low-level PSP pathology that clearly wasn't enough to manifest with symptoms during life. So this is telling us that actually the assay is working really well. It's picking up even low-level PSP pathology. So this is telling us that even in the very earliest stages of the condition, this test might be accurate enough to pick up the diagnosis. So now our attention is currently on getting the tau assay to work in spinal fluid samples from living PSP and CBD patients. And this work is currently ongoing. So I really do urge you to watch this space. We hope to have some exciting, promising results to report in the not so distant future. In terms of other work, we're also very keen to explore whether these seed amplification assays can be used not only to give us a binary positive or negative result, but perhaps certain quantitative measures from the aggregation curve, like the lag phase or the total level of fluorescence that we see in the aggregation curve, whether these quantitative measures might be markers of how quickly or slowly the pathology is spreading in the brain. And this may in turn correlate with how quickly or slowly a patient is progressing clinically. And if that's the case, it would suggest that these tests may have an additional role, not only as a diagnostic test, but also as a prognostic test, as well as being used in a clinical trial as an outcome measure. So potentially multi-use of these tests. So we're really excited about the progress being made in this project, and clearly there's a lot more to come. And none of this would be possible without, first of all, the funding from the PSP Association, but also the fantastic colleagues and collaborators that I work with. So a big thank you to them. And also thank you very much to you in the audience for listening. I'm very happy to take questions.
Thank you, Ed. I found that very interesting, exciting, and presented very clearly, although I think many of us will probably want to watch it again. So I'm not seeing any questions online, Megan. Oh.
So there was one question that came through from Tom, and it says, what function does normal tau play in the brain?
Yeah, it's a really good question. So the normal function of tau is that it stabilizes these structures called microtubules in nerve cells. And we know that in the setting of PSP and CBD, the tau isn't doing its job. It becomes abnormal and can't do its job at stabilizing those microtubules within nerve cells. And that eventually causes those nerve cells to not function properly and then eventually die off. So that's the normal physiological function of the tau protein.
Thank you. Other questions for Ed?
There was one that came through in the chat directly to me from Suzanne, and it said, how relevant are increasing double vision and balance issues to brain degenerational disease?
Yeah, so as we know, unfortunately, one of the main features of PSP is this difficulty with eye movements. And that can manifest clinically with double vision, difficulty focusing, difficulty looking up and down. And the cause of that is this actual tau pathology that affects a specific part of the brain called the midbrain. It's unsurprisingly in the middle of the brain. And that's the part of the brain that controls our vertical eye movements. And that's why PSP patients suffer from this difficulty in moving the eyes up and down and focusing.
Thanks, Ed. I've just gone back. I've seen an earlier question from Amanda that you may well be able to comment on. Do we yet know if subtype can change with disease progression in terms of initial diagnosis versus pathological diagnosis? And if so, how?
So I think what the question is referring to is these different subtypes of PSP, the different clinical subtypes of PSP. Yeah. So as I mentioned in my own talk, we know that broad main subtypes of PSP, there's the classical form, which we refer to as Richardson syndrome. And this is the classical form where patients will have very early balance problems, eye movement problems, speech and swallowing problems. And then the second form is this subcortical form of PSP that looks really a lot like Parkinson's disease, very difficult to distinguish in the early stages of the condition. And then the third subtype is more the cortical form of PSP, where there's more of a burden of cognitive symptoms, cognitive and behavioral symptoms early on in the condition. Now, these different subtypes, they differ in the early stages of the condition, so usually in the first 3 years of symptoms. Whereas what we've found in the prospect study is that as each of these different subtypes progress towards the latter stages, they all look like Richardson syndrome. So yes, to answer your question, it's not uncommon at all for someone to have one subtype, especially if it's the alternative forms of PSP, and then for that later down the line to convert and look more like the classical Richardson syndrome towards the latter stages of the condition.
Thanks, Ed. Right. Megan, any more questions?
One more that's just come through. I don't know if you'll be able to answer this, Ed. So it's quite a general question of how close are researchers to developing a medication to help with PSP?
Yeah, that's a frustrating question to answer, Megan, because as you know, unfortunately, we don't have any effective medications at this point in time that can stop or slow down the progression of the disease itself. And the other big frustration is that in contrast to Parkinson's disease, where again, we don't have any effective medications to stop or slow down Parkinson's disease progressing, but what we do have are symptomatic treatments that can aid with improving the symptoms of tremor or stiffness or slowing down. And one of the biggest frustrations is that those symptomatic treatments don't seem to work as effectively in PSP. So there's a lot of focus on both fronts, both the development of symptomatic treatments that can help with the symptoms of PSP, whether they be movement problems or the non-movement problems, the problems with mood, apathy, these problems as well. These are very important symptoms to tackle, but also the big focus of research, in particular what we're doing at UCL, is use of fluid biomarker research avenues that can potentially slow down or stop the disease in its tracks. So there's a very active area of research, and we hopefully will be able to find some new treatment avenues in the not so distant future.
Thanks, Ed. Right, a question from Siobhan. Has there been any research into the use of hydrogen therapy to help reduce the symptoms of PSP?
Not that I'm aware of. I'm not familiar with hydrogen therapy at all, I'm afraid. Sorry.
Okay. Well, I think we've gone through the questions, so I will wrap it up there. I would just like to say a big thank you to all the speakers who've shared their latest research with us today, and also to all of you for spending time listening in, asking your questions. And just to remind you that we do have a recording, so we will share the link with everyone so that you can review it again and pass it on to anyone you know who couldn't make it today. So with that, I will wrap us up 15 minutes early, and yes, a big thanks to all of you.